Good afternoon. My name is Irene. I'm working at Sanger Institute. I am advanced research assistant, working in long read team. And I'm going to work with you with the no experience people in Arima, one of the person with Morten and with Karine. And we are going to talk a little about what is our schedule in the week. And then Morten's going to talk a little more about that. Let me see. Oh, this is the schedule you have there. Everything. This is Sunday. We're going to work here, but tomorrow we will start with all the talks. In the morning, we should have the dovetail talk, Arima protocol. Then we have a coffee break. We have to be sure you have everything in the folder, right? But we have to be very concentrated in the process because it's very tight, the time. The first day, Monday, is very, I mean, we have to spend time to learn the process, but the time it can be a little difficult for tomorrow. So, and then it should be more easy because we have a long period of incubation in Arima. So we will have more maybe time relaxed to be around and to wait a little. But the first day, we are going to learn something that is very important for HiC, that is weight the tissue, also to grinding the tissue and prepare the tissue for start the process. So it's a very important part of the HiC Arima, the fixation of the tissue, is that we need to fix the chromatin and collect the exact 3D position of the chromatin. So that is very, very important. So we need to be, we need to work together to be there. And then Tuesday we have the lysis, we have the cross-linking. This is going to be Tuesday, sorry. And then on Wednesday, we will work in the lab until lunch, and then we'll start with several other discussions. And that is very important because I think with Karine, I agree with Karine that we are not expert in Arima because there are a lot of kind of tissue that we can process. We have in Sanger a lot of different tissue because we work with the Tree of Life project with Graeme, but even that is impossible. And we have several problems, and the discussions are going to be very good for us and to share the knowledge that we collect in all of these years, months.
So the difference between these two procedures is that for the dovetail or the cantata, I probably want to say that through the whole week. It's a bit quicker to do, and it's the only one that we could actually finish with a library at the end of this course. The Arima protocol, which is more time-consuming, will give us the proximity ligated DNA, which then has to go into the final library prep. It's a time issue, and that's what we can fit for this week, especially if you want to go into details on different samples, etc., and troubleshooting. But it's interesting to see the two different approaches. The assembly link.
There. So all the libraries, Arima libraries, will be prepared in the coming couple of weeks. And then we'll sequence them, QC sequence, all libraries that you prepared for during the summer, and we'll feed you back afterwards. So all that will not be lost. So you will get all the feedback back sometimes in August or September.
Yeah, and we'll probably set up some Zoom meetings, I guess, to go through all the results, so we are on the same page in the end. The dovetail is kind of similar. It starts with cross-linking the tissue, and then you go directly into a cutting or tegmentation reaction, which kind of makes your DNA or proximity ligated DNA at a suitable size for sequencing already. So you're kind of fast forwarding a bit compared to the Arima. There are some advantages and disadvantages of this, but we can come back to that later. On Tuesday, where we are actually QCing how well did this cutting of the DNA go. And then for the rest of the Tuesday is kind of packed with different incubation reactions to get the final library. And hopefully we have a final library that we can show. I don't know what we are doing if we need to redo the lysate thing, but we can talk about that if it happens. And the thing is, we have mammal DNA, tissue, I mean, so it should work.
Yeah.
So, yeah. Okay.
Okay, right. There is some recommendation that we have because we have to work with formaldehyde in HiC, Arima, probably in dovetail too. Yeah, okay. And also we are going to work with the liquid nitrogen. I don't know if everybody worked already with that, but I put here some recommendations, general recommendation, but anything else that happened, we have to contact the organization of the group, because every of us are from different labs, and we don't know how the lab works here, right? How they manage that. But we are going to be, of course, with Olga and with all the people from here. But we have the picture already of the film field that we need, a special to everything. Everything must be done in the ducted fume cupboard for formaldehyde. We need a special bottles. If you have any, split in this, let us know, because any other person can continue to work if we have something in the cup, in the fume. Okay, just let us know so we can clean or do wherever the lab procedures have to do. The gloves are very important. If you know, probably you know already, the orange glove we're going to use here. In Sanger, we use a different that is a little more like a more resistant, probably. It's a blue gloves. And it's very uncomfortable, to be honest. But so the orange it should be super fine and very good to protect. And what else? Well, of course, the lab coat that we are going to have. This is the health hazard of the formaldehyde in general. Probably we will leave the presentation for that, right? Yeah, right. So you will have everything here. But I assume that everybody here that work in molecular biology knows how to proceed with this kind of liquids. And about the liquid nitrogen, it's important to use a correct PPE. We are going to have that in the course. Some in general, some people to extract from the big bottle use the face shield, the gloves, different gloves to extract the liquid nitrogen from the big bottle. And then we have a small bottle in the lab probably, yeah. And proper clothes, of course, to be in the lab, proper shoes. And probably we're going to have in the lab, it's recommended to have the oxygen depletion alarm when we are going to work for a big, not big, but a period with that liquid nitrogen. So they are going to tell us if we need more oxygen in the room. So yeah, that's some of the problem that we have. Can cause the liquid nitrogen. Sometimes you really don't think that nothing happened, but it has happened already. So you have to be very protected with this, especially the people that are going to work with the liquid nitrogen directly. But Morten, Olga, everybody is going to be there to help if you never use it. And if you never work with liquid nitrogen, let us know so we can show from the beginning how to use. Yeah. So for Arima, the first part, we need to do the tissue weighing and the grinding. That means that we have to have the correct amount of the tissue to start the process. And we need to process with the liquid nitrogen to have powder if it's possible to have the correct reaction in the end. Well, the input, I'm going to talk a little about the Arima HiC in general. Morten just told me that there is a new protocol that we are going to check in the end of the day. So tomorrow we are going to have the very new version, but it's not going to change a lot. So this is, and then I'm going to talk a little about what we do in Sanger, because it's a little different. So the input, it should be fresh, frozen animal tissue. Something that we notice that is very important is the tissue has to be frozen all the time. You never can open the box. For us, of course, I'm talking from Sanger. You never can open the box on the bench without dry ice. Never. This is very important to maintain the tissue in frozen. That's very important. So if you are going to pick up your samples, it's very important to have next to you dry ice, because the samples should be -70. The output, we need powder of this tissue to start the cross-linking with the formaldehyde. So Arima worked with 50 to 200 milligrams of fresh frozen tissue. And then less than 50 milligrams, they consider like a low input protocol. The protocols change a little. So we have a standard protocol of Arima HiC and a low input protocol. And then we have a different others protocols for plants, for nematodes, for different things that we have to change for this, especially in the first day, that is the cross-linking, right? So then the samples have to make like a powder with the liquid nitrogen. I think Karine is the expert on that. Yeah, well, the people from Genoscope, both of them, they work a lot with that. We in Sanger, for our situation, is different, and I'm going to talk a little about that. So it's important to have a powder, right? It's very important to have a powder. The cross-linking we prepare at the fume hood because we work with formaldehyde. So that is the first step. And the first step is already with the formaldehyde, and we are going to work in the correct place. So for Sanger, for us, we make a different change. We know the expert, Mike Wild. He was adapting the protocol for us because we are in a Long Read is a production team. So we need to have everything, a protocol that we can use to do in a production, to do a lot of samples, right? So we process normally one person, 16 samples a week, but normally I have 4 people working with 16 samples, depend of the week, depend of the month. So we need a protocol suitable to do in a lot of quantity, right? And also the amount, Mike changed. We call a standard protocol the protocol that Arima calls low input. So we work with 50, normally 30 milligrams of tissue. We work with that, and that is our standard protocol. My colleague, Graeme, is from Tree of Life, the group of Tree of Life, and their group, his group prepared the tissue for us. So we receive already normally the tissue that was weighted with the correct amount. So we process. Sometimes we receive already the powder, sometimes a little piece of tissue, and sometimes we receive empty tubes. Empty tube, but it's not empty. It's very ultra low. So we process something that actually we can't see, never. We just see the results in the end. So that is very tricky, and I'm going to talk a little about that. We still work in that process because we are receiving feedback from the expert that work in the bioinformatician part and whatever, but it's very complicated. So it's going to be a discussion that we're going to have on Thursday. So yeah, we can hear suggestions and everything, but yeah. And so we have, depend of the tissue that we are going to work, we use different process, cryo prep. We use pulverized or pellet. We receive a pellet. They also work with fast prep, right? The fast prep and with different bit beaten. So it's it's a process that they construct already, right? It's something that they were study and how can I say, study and testing all the time. And depend of the tissue and depend of the end of the product, we can start to use that. So it's a very nice project because you always have something new. But our protocol is something like that. We have several other things where plant protist and all of has numbers, right? HiC protocol 2, 3, 4, and some of them we are still working. I'm working normally here with nematodes and targeted. Ultra low input is my my responsibility right now, but it's still complicated. We are receiving different feedback and yeah, it's something that is still difficult to work. What else? Well, this is something that we always, when we receive something new, we have to check with Mike because he started to apply the HiC protocol at Sanger. But now, because we have a lot of samples, sometimes we check with Mike and he said, I never processed that, so maybe we can try with this. So there is a lot of different issues. We are going to use I'm going to make a demonstration of how we process the samples, because we agree that during the course, everybody has to work with liquid nitrogen. I mean, every group that we are going to have, you are going to do that part by yourself and with the teacher. And I'm going to do a demonstration with this. Is the power measure. We have these elements. We receive the tubes we can receive in a Eppendorf 1.5 or in a special FedEx tubes with code and everything of the sample. And we transfer the sample to special tubes. If you can see, we have here a little, it's different, right? Here is the tube, normal tube, and here we have some kind of, the texture is different, and that is important because with this vessel, we introduced, and with a little instrument, we just
It's a gun.
Oh yeah, it's a gun. Exactly, yeah.
I checked in the baggage for some reason. Just to be on the safe side.
Yeah.
So this is it. I mean, it's just a simple drill, basically. And it has this tube. The tube has this kind of pattern on the inside, which fits very well with this vessel or whatever it's called.
So yeah, you can see the tube that is completely different at some point, and this is very useful. We started to use a 1.5 Eppendorf in the beginning of HiC, but this is much better to process a sample. And what else? Something already important, because I'm not going to work with all the groups. We are going to be split, of course, and I'm going to tell my group what is the importance of this part for us. We saw that in a different extraction, when we use this instrument, this gun, we started to process like a 1 minute, something like that. And if you work with long read, probably you are damaging your DNA, cutting your DNA, whatever. So in the other extraction, they recommend to don't do that, right? But in HiC, we never heard anything, bad feedback, but we decided to just make the power mashing only maximum 30 seconds. Sometimes if you have already a cloudy liquid in 10 seconds, that's it. Don't do more, because you are damaging your DNA. This is for long read, but for us, we thought that maybe is a very important detail. So why I'm saying that? Because in the past, we process with power machine for a minute sometimes, because you want the DNA, right? You wanted to extract all of DNA. You can see that little tissue there, and you said, I have my DNA there. So no, it's not good. If you have already cloudy, and if you still have some part, just leave it. Just to obtain the size, the correct size that you want. Another thing is Claudia. Yeah,
Was that the long read you meant?
Long read.
Okay.
Yeah. For the short read, it should be fine.
So for this purpose, for the high C, you're not afraid of doing 2 minutes.
Yeah. Yeah, no, 2 million is too long. Yeah. Okay. And this is for our standard protocol, right? Because for us, we receive a lot of DNA, right? So it's not needed to do for a long time for us. We saw that because you have very soon a cloudy liquid. So you power mash in the tissue already. I think that's it for me. Yeah. Yeah.
But you do the power master in liquid, right?
Yes. It's the MPBS 1X. Yeah. And yeah, only PBS. Yes.
And then how do you decide which of your different samples, which of those, cryo prepping or power masher or?
Yeah. Well, this is something that is a Graeme group normally do. How do you decide to do one or the other process?
Well, we're based on the weight of our initial sample. For plants, we always would fast prep or cryo prep things, so we sort of grind down into a fine powder. A lot of the sort of insects will be smaller than, I think, 50 milligrams. So if a sample is, say, 50 to 60 milligrams, we'll cryo prep it. And if anything smaller than that, we'll just have to power mash.
Yeah. Well, it's but basically it's in the weight. Yeah. Okay.
One other thing.
Yeah.
You cross-link before you do it, right? Or on some of the samples, low input samples, or do you always cross-link after the crushing?
We cross-link it first, and
Yes.
then we
For that one, yeah.
for HiC protocol, and then we went to the lab, not in the fume hood, and we power measure in the lab with suppose that is everything fixed up. Right. Yeah. You are going to understand better. The people that doesn't have experience are going to understand very good tomorrow.
You understand what we mean by the high C, what's the purpose of this, right? That we start from tissue, not DNA. We want to conserve the structure between the DNA and the chromatin. Yeah. Capture the 3D structure within the chromosome, which are then kind of separated into different compartments they call A and B, which is active, inactive regions, which then means are divided into smaller topologically associated domains, that they call them pads for short, which also then has these shorter chromatin loops, which we want to cross-link and capture. So we can see that there are connections within the chromosome, as well as between chromosomes, interchromatin. So that's the whole idea behind it. And all of the things we are learning now during this week can be utilized on other fields of gene regulation space, to put it like that. Because it's the same techniques, basically. They're used in human medicine to find fusions of genes, for instance, and other regulatory aspects that they are interested in. So how enhancers are influenced in the 3D space to connect to promoter regions tells us something about how gene expression is regulated. And that was the primary source for developing these techniques to begin with. You had this 2C to 3C, which is chromatin conformation capture, and then you even have 4C and 5C, I think, and then it ended up just being high C, because there you capture everything, whole genome wise. That's the idea behind it. Okay,